Namaskar, welcome back to the lecture series. In this episode, we are going to see the large scale synthesis of recombinant proteins. There is no doubt that the production of recombinant proteins in microbial systems has revolutionized biochemistry. The days where kilograms of animals and plant tissues or large volumes of biological fluids were needed for the purification of small amounts of given proteins are almost gone. The ability to express and purify the desired recombinant proteins in a large quantity allows for its biochemical characterization, its use in industrial processes, and the development of commercial goods. At the theoretical level, the steps needed for obtaining a recombinant protein are pretty straightforward. You take your gene of interest, clone it in whatever expression vector you have at your disposal, transform it into the host of your choice, induce, and then the protein is ready for purification and characterization. In practice, however, dozens of things can go wrong. Poor growth of the host, inclusion bodies, formation, protein inactivity, and even not obtaining any protein at all are some of the problems often found down the pipeline. Yet, in the fields of recombinant protein expression and purification, progress is continuously being made. Let's see how the organisms to use. The choice of the host cell whose protein synthesis machinery will produce the precious protein will initiate the outline of the whole process. It defines the technology needed for the project, be it a variety of molecular tools, equipment, or reagents. Among the microorganisms host systems that are available include bacteria, yeast, filamentous fungi, and unicellular algae. All have strengths and weaknesses, and their choice may be subjected to the protein of interest. For example, if eukaryotic post-translational modifications like protein glycosylation are needed, a prokaryotic expression system may not be suitable. Mostly E. coli is used as a special system. The advantages of using E. coli as the host organism are very well known. It has unparalleled fast growth kinetics in glucose salt media and given the optimal environmental conditions. Its doubling time is about 20 minutes. However, it should be noted that the expression of a recombinant protein may impart a metabolic burden on the microorganism, causing a considerable decrease in generation time. Secondly, high cell density cultures are easily achieved. Thirdly, rich complex media can be made from readily available and inexpensive components. Finally, transformation with exogenous DNA is fast and easy. Plasmid transformations of E. coli can be performed in as little as 5 minutes. Let's see about the plasmids chosen. The most common expression plasmids in use today are the result of multiple combinations of replicons, promoters, selection markers, multiple cloning sites, and fusion proteins. Fusion protein removal strategies are also been illustrated in figure 1. The figure 1 indicates the anatomy of an expression vector. The figure depicts the major features present in common expression vectors. All of them are described in the text. The affinity tags, the coding sequences for their removal were positioned arbitrary at the end terminus for simplicity. Multiple cloning sites and coding sequences of the desired proteins were also indicated. Coming to replicon, genetic elements that undergo replication as autonomous units, such as plasmids, contain a replicon. It consists of one origin of replicon together with its associated cis-acting control elements. An important parameter when choosing a suitable vector is copy number. The control of copy number resides in the replicon. It is logical to think that high plasmid dosage equals more recombinant protein yield as many expression units reside in the cell. However, a high plasmid number may impose a metabolic burden that decreases the bacterial growth rate and may produce plasmid instability, and so the number of healthy organisms for protein synthesis falls. For this reason, the use of high copy number plasmids for protein expression by no means implies an increase in the production yields. Commonly used vectors such as PET series possess the PMB1 origin, while a mutated version of the PMB1 origin is present in the PUC series. The wild type COL-E1 origin can be found in the PQE vectors. They all belong to the same incompatibility group, meaning that they cannot be propagated together in the same cell, as they compete with each other for the replication machinery. For the dual expression of the recombinant protein using two plasmid systems with a P15A origin are available, though rare, triple expressions can be achieved by the use of PSC101 plasmids. This plasmid is under a stringent control of replication, thus it is present in a low copy number. The use of plasmid bearing this replication can be an advantage in case where the presence of high dose of a clone gene or its product produces a deleterious effects to the cell. Alternatively, the use of the dual vector simplifies the dual expression by allowing cloning of the two genes in the same plasmid. The dual vectors possesses two multiple cloning sites, each preceded by T7 promoter, a LAC operator and a ribosome binding site. By combining different compatible duet vectors, up to 8 recombinant proteins can be produced from 4 expression plasmids itself. Let's see about a promoter. The staple in prokaryotic promoter research is undoubtedly the LAC promoter. Key components of the LAC operon. Lactose causes induction of the system, and this sugar can be used for protein production. However, induction is difficult in the presence of readily metabolizable carbon source. If lactose and glucose are present, expression from the LAC promoter is not fully induced until all the glucose has been utilized. At this point, low glucose cyclic adenosine monophosphate CAMP is produced, which is necessary for complete activation of the LAC operon. This positive control of expression is known as catabolite repression. In accordance, CMB levels are low in the cells growing in LAC operon repressing sugars, and this correlates with lower rates of expression of the LAC operon. Also, glucose abolishes lactose uptake because lactose permease is inactive in the presence of glucose. To achieve expression in the presence of glucose, a mutant that reduces sensitivity to catabolite regulation was introduced, the LAC UB5 promoter. Basal expression control can be achieved by the introduction of a mutated promoter of LAC-I gene called the LAC-IQ that leads to higher levels of expression almost tenfold of a LAC-I. The LAC promoter and its derivative LAC-UV5 are rather weak and is not very useful for the recombinant protein production. Synthetic hybrids that combine the strength of other promoters and the advantages of the LAC promoter are available. The T7 promoter system present in the PET vectors is extremely popular for recombinant protein expressions. In this system, the gene of interest is cloned behind a promoter recognized by the phase T7 RNA polymerase T7a RNAP. This highly active polymerase should be provided in another plasmid, or most commonly it is placed in a bacterial genome in the prophase lambda DE3 and coding for the T7 RNAP under transcriptional control of LAC-UV5 promoter. Thus, the system can be induced by the lactose or its non-hydrolyzable analog isopropyl beta D1 thiogalactopyranoside IPTG. Basal expression can be controlled by LAC-IQ but also by T7 lysozyme co-expression. T7 lysozyme binds to T7 RNAP and inhibits transcription initiation from the T7 promoter. In this way, if small amounts of T7 RNAP are produced because of leaky expression of its gene, T7 lysozyme will effectively control unindented expression of heterologous genes placed under T7 promoter. T7 lysozyme is provided by compatible plasmids called the P-lysis S or P-lyse. After induction, the amount of T7 RNAP produced surpasses the level of polymerase that T7 lysozyme can inhibit. The free T7 RNAP can thus engage in transcription of the recombinant gene. Yet, another level of control lies in the insertion of a LAC-Q operator downstream of the T7 promoter, making a hybrid T7 or LAC promoter. All three mechanisms, tight repression of the LAC inducible T7 RNAP gene by LAC-IQ, or T7 RNAP inhibition by T7 lysozyme, and presence of LAC-O operator after the T7 promoter, make the system ideal for avoiding basal expression. Another widely used approach is to place a gene under the control of a regulator phase promoter. The strong left-forward promoter PL of the phase lambda directs the expression of early lytic genes. The promoter is tightly repressed by the lambda CI repressor protein, which sits on the operator sequences during the lysogenic growth. When the host SOS response is triggered by DNA damage, the expression of the protein REC-A is stimulated, which in turn catalyzes the self-cleavage of lambda CI, allowing transcription of PL control genes. This mechanism is used in the expression vectors containing the PL promoter. The SOS response and the recombinant protein expression can be elicited by adding nalidixic acid, a DNA guidase inhibitor. Another way of activating the promoter is to control lambda CI production by placing its gene under the influence of the another promoter. In the PLX series of vectors, the lambda CI repressor gene was integrated into the bacterial chromosome under the control of TRP promoter. In the absence of tryptophan, this promoter is always on and the lambda CI is continuously produced. Upon addition of tryptophan, a tryptophan TRP-R repressor complex is formed that tightly binds to the TRP operator, thereby blocking lambda CI repressor synthesis. Transcription from all the promoters discussed so far is initiated by the chemical cues. Systems that respond to physical signal are also available. The prial promoter is one example. Selection marker. To deter the growth of plasmid-free cells, the resistant marker is added to the plasmid backbone. In the E. coli system, antibiotic resistant genes are habitually used for this purpose. Resistance to ampicillin is conferred by the BLA gene, whose product is a perisplasmic enzyme that inactivates the beta-lactam ring of beta-lactam antibiotics. However, a beta-lactamase is continuously secreted, degradation of the antibiotic ensures, and in a couple of hours ampicillin is almost depleted. Under these situations, cells not carrying the plasmid are allowed to increase in number during the cultivation. For this reason, tetracycline has been shown to be highly stable during cultivation because resistance is based on active efflux of the antibiotic from the resistant cells. Different subtypes of plasmid addiction systems like toxin, antitoxin based system, metabolism based system, and operator and repressor titration systems. While this promising technology has been proved successful in large scale fermenters, expression systems based on plasmid addiction are still not widely distributed. Let's see how the affinity tags issue goes here. When devising a project where a purified soluble active recombinant protein is needed, as is often the case, it is invaluable to have means to detect it along the expression and purification scheme, attain maximal solubility, and finally easily purify it from the E. coli cellular milieu. The expression of a stretch of amino acid, a peptide tag, or a large polypeptide fusion partner in tandem with the desired protein to form a chimeric protein may allow these three goals to be straightforwardly reached. Being small, peptide tags are less likely to interfere when fused to the protein. Vectors are available that allow positioning of the tag on either at the N terminal or the C terminal end. If the three-dimensional structure of the desired protein is available, it is wise to check which end is buried inside the fold and place the tag in the solvent accessible end. Common examples of polypeptide tags are poly-arch, flag, FLAG, poly-hitch, C-MIC, and S and strep2 tags. Since commercial antibodies are available for all of them, the tagged recombinant protein can be detected by Western blot along the expression tiles, which is extremely helpful when the levels of the desired protein are not high enough to be detected by the SDS page. Also, tags allow for one-step affinity purification, as resins that tightly and specifically bind the tags are available. For example, his tag proteins can be recovered by immobilized metal ion affinity chromatography using nickel and cobalt loaded nitrolo triacetic acid agarose resins, while anti-flag affinity gels are used for capturing flag fusion proteins. On the other hand, adding a non-peptide fusion partner has the extra advantage of working as solubility enhancers. The most popular fusion tags are the maltose binding protein and utilization substance protein A, which is called as NUSA, thioredoxin TRX, glutathione S transferase GST, ubiquitin and SUMO. NUSA, BMP and TRX display the best solubility, enhancing the properties, but their large size may lead to the erroneous assessment of the protein solubility. Indeed, when these tags are removed, the final solubility of the desired product is unpredictable. For these reasons, smaller tags with strong solubility enhancing effects are more desirable. Recently, the 8-cadre calcium binding protein of FH8 from the parasite Fasciola hepatica was shown to be as good as or better than the larger tags in terms of solubility enhancement. Moreover, the recombinant protein maintained their solubility after the tag removal. A different group of fusion tags or stimulus responsive tags, which reversibly precipitate out the solution when subjected to the proper stimulus. The addition of beta role tags to a recombinant protein allows for its selective precipitation in the presence of calcium. The final products presented a high purity and the precipitation protocol only takes a couple of minutes. Another protein based stimulus responsive purification tags or elastin-like polypeptides, also called as ELPs, which consists of tandem repeats of the sequences like VPGXG, where X is valine or alanine or glycine in 5:2:3 ratio. These techniques represent an alternative to conventional chromatography based purification methods and can save production costs, especially in the large scale settings. The main characteristics of the tags mentioned in this section are outlined or illustrated in table 1. Let's see how these tags are removed. For the section tag removal, if the structural and biochemical studies on the recombinant protein are needed, then the fusion partner must be eliminated from the recombinant protein. Fusion tags should be removed too because they can interfere with the protein activity and structure, but they can be left in their place even for crystallography studies. Tags can be eliminated either by enzymatic cleavage or chemical cleavages. In the case of tag removal of enzyme digestion, expression vector possesses sequences that encode for protein cleavage site downstream of the gene coding for the tag. Enterokinase, thrombin factor XA and the tobacco H virus TEV protease have all been successfully used for removal of peptide tags and fusion partners. As the name implies, in chemical cleavages, the tag is removed by the treatment of the fusion protein with chemical reagent. The advantages of using chemicals for this purpose are that they are easy to eliminate from the reaction mixture and are cheap in comparison with the proteologic enzymes, which makes them as an attractive choice in the large scale production of recombinant proteins. However, the reaction conditions are harsh, so their use is largely restricted to purified recombinant proteins obtained from the IBS. They also often cause unwanted protein modification. The most common chemical cleavage reagent is cyanogen bromide CNBR. Cyanogen bromide cleaves the peptide bond at C terminal to methionine residues, so This amino acid should be present between the tag and the protein of the interest. Also, the target protein should not contain internal methionines. CN cleavage can be performed in common denaturing conditions with 6 millimolar gonadine chloride or 70% formic acid or trifluoroacetic acid. Now let's see about suitable hosts. For the first expression screen, only a couple of E. coli strains are necessary. BL21DE3 and some derivatives of K12 lineage like other parental B strains, BL21 cells are deficient in lone protease, which degrades many foreign proteins. Another gene missing from the genome of the ancestors of BL21 is the one coding for the outer membrane protease OMPT, whose function is to generate extracellular proteins. The liberated amino acids are then taken up by the cells. This is problematic in the expression of a recombinant protein as after cell lysis. OMPT may digest it. In addition, plasmid loss is prevented thanks to the HSD-SB mutation already present in the parental strand B834 that give rise to BL21. As a result, DNA methylation and degradation is disrupted. When the gene of interest is placed under a T7 promoter, then T7 RNAP should be provided. In the popular BN21DE3 strain, the lambda DE3 prophase was inserted in the chromosome of BL21 and contains the T7 RNAP gene under the lag UV5 promoter. The DL21DE3 and its derivatives are by far the most used strains of the protein expression. Still, there are reports where the K12 lineage is used for its purpose. A D494 and the origami strains Atrix B mutants, so disulfide bond formation in the cytoplasm is enhanced. Another widely used strain from the K12 report here is HMS174, a Rickia mutant. This mutation has a positive effect on the plasmid stability. At this point, it should be pretty clear that the number of options when designing an expression system is considerably high. Choosing the perfect combination is not possible as a priori, so multiple conditions should be tested to obtain a desired protein. If the project demands expressing 2 protein constructs cloned in 6 different expression vectors, each transformed in 3 different expression strains, then you are in for 36 expression trials. This number may even be higher when other variables are taken into account. This trial and error and the time consuming pilot study can be made faster if micro expression trials were performed before scale up. Small scale screens can be performed in 2 ml tubes or 96 well plates. High throughput protocols adapting automatic liquid handling robots have been described, making it possible for a single person to test more than 1000 cultures within a week. Here we come to an end of this large scale synthesis of recombinant proteins. Hope you enjoyed the session and thanks for watching.